All this is Dr. Mobeen Sayed from Drbeen.com. Welcome to one more show. So the discussion today is something that this is one study that has been disturbing me, I think, since it was published in December 2023. I want to discuss this. The study basically says the following. It says that messenger RNA-based vaccines, and it is not necessarily just the COVID vaccine, they ran this test on the COVID vaccine, but any mRNA-based product, vaccine-like product, causes ribosomal slippage or makes bad proteins, makes proteins that it is supposed to make, but these are mistargeted, off-targeted, broken proteins. These broken proteins can actually look like our tissue proteins, causing our immune system to attack our tissues and produce autoimmunity. And then there is a further problem, and this is me not study, this much is study what I said before. Once the immune system starts attacking our cells and breaking them open, from within the cell, the guts of the cell that spill out, that is the mitochondria and the nucleus and enzymes and other proteins that are usually packed within the cell, when they become exposed, our immune system starts attacking them as well. And all of a sudden, this is called autoimmunity escalation. All of a sudden, we have autoimmunity escalation. So it is possible that in some people, messenger RNA vaccines will produce autoimmunity. Then the immune system, as it attacks the cells, will produce autoimmunity escalation, creating a long-term autoimmune disease. And this has been disturbing me for a long time. The reason for that is that whenever I see someone, as rare as it may be, the vaccine injury from mRNA vaccines, whenever I see someone who is autoimmune, whose autoimmunity has become escalated, I think about this study. And this study has an interesting closure. And that is that the authors from Cambridge and Oxford and Liverpool universities, they actually offered a solution. They said, do this and this will not happen. And I do not believe that that solution has been implemented. So what I wanted to do today is, for anyone who is vaccine injured and maybe stuck in an autoimmune state, at least I wanted to present to them that what could be one of the reasons. There are so many other reasons that are spike related, but this is an off target, a bad protein and cross reactivity related problem. Now, could this happen because of long COVID as well? Interestingly, long COVID has not known to produce this kind of a protein. Similarly, the non-mRNA vaccines, because they do not have modified synthetic mRNA in them or nucleotides in them, they also do not produce this kind of a problem. So the researchers actually compared the Pfizer-BioNTech vaccine to Oxford AstraZeneca vaccine, and they found that the BioNTech produced this problem and AstraZeneca, which is a non-mRNA vaccine, did not produce this problem. So as much as it is fascinating to see this kind of a mechanism, it is more important to understand why autoimmunity may be occurring in some people and then escalating and becoming more and more a problem for them. Now, can this be then solved? Yes. When you sit down with your immunologist, there is going to be a discussion about how to bring the immune system back towards normal. You may remember a few days ago, I did the study where for the first time, they showed a signature of immune abnormality in vaccine injured individuals where the T helper cells were less and the B cells were more. And the B cells were stuck in an unswitched state. And the T cells were so much working that they had become exhausted as well. So they were a mixture of the active and exhausted T cells. And there was a problem with the innate arm. So if you take that study, and if you take this study and you sit down with your immunologist, you can actually figure out what is the way to move forward. There are drugs like PD-1L and there are drugs like CTLA-4 drugs, which can suppress the autoimmunity and give some level of comfort and some healing, at least some suppression of the symptoms. So with this, thank you very much for joining me on this one. This study I had actually done once before as well in 2023. And I'm repeating this because this actually keeps coming back to my mind every time I see somebody who is suffering. So let's look at it together. So this is drbeen.com. If you would like to have more lectures, just become a member of drbeen.com. The links are in the description. Plus all the studies that I'm showing and more references are in the description of this video. You can just go to the description. Many times people say that, hey, where is the link? My links usually are in the description of this video. Okay, so this is the write-up about the study. This is University of Cambridge. If you look at the date here, 6 December 2023. This is a write-up which is interesting. I'll discuss that in a second. This is the study itself, printed in Nature, published, accepted 6 December 2023. N1 methyl pseudouridylation of mRNA causes +1 ribosomal frame shifting. Such an important problem. I think it may be the basis of lots of vaccine related adverse events. And I do not see a reaction to this. So either share my video with someone who may be able to do something about it, or send them the links from the description of my video. So having said that, let's look at my diagrams and the discussion. So the one concept that I would like you to keep in mind throughout learning this study is molecular mimicry, and then the related concept is cross reactivity, which will then lead to autoimmunity, which will then lead to escalated autoimmunity. So this is a chain of 4 items that if they stay in the top of your mind, then this study would start making more sense. And it would also start making sense that why some people are stuck with the vaccine side effects for years now. So gifts for humanity. Here is the basic idea that imagine that the vaccine wanted to make this spike protein, but because there is a problem that the nucleotide are slipping on the ribosomes, and I'll explain that a little more in a second, the resulting protein could end up being an off-target protein, as they're calling it, or a truncated protein. Now, I want to explain one important concept over here. That concept is this, that when you have, imagine that you have a long thread of a protein. This long thread is actually made up of amino acids, which are the building blocks of the proteins. These amino acids have, so let me just make them, let's say, various colors here. So every color represents a different amino acid. And there are 20 or so amino acids that are used in our body. Now, each amino acid has some chemical properties which makes it positively or negatively charged or neutral even, and then have a dent in it or a bend in it. These amino acids and their charges, like little magnets, they attract each other. The result of that attraction is that this thread of the protein becomes folded into a 3-dimensional structure. So imagine that you have a long string of magnets, and as soon as you leave them on the floor, these magnets start attracting each other, and slowly they turn into a shape. So when the spike protein amino acids put together in that one string and you allow it to become a shape, it makes the spike protein. But if you take a part of that structure, imagine just this much part, then it is not necessary that the resulting protein is going to be the initial part of the spike protein. That's not necessary. Do you know why? Because imagine that this is the part that is going to be made, or the amino acids for this part are present. The remaining amino acids are not working. And why not? We'll see. This thread here will fold in totally different ways because the electromagnetic and chemical bond forces are different here. So it is not necessary that this thread would make the initial part of the spike. It actually might end up being a very different protein folded. And that very different protein might actually be a protein that would mimic, that would look like a protein on our cell's surface or our tissue surfaces. And when the immune system would attack this protein, the same immune system is now going to start attacking our cells. And when that cell would break open because of the immune attack and the things would come out, more attack would occur. And now we have autoimmunity and escalated immunity. So continuing on, here is what they said. They said, frame-shifted residues were not included. We found that response to +1 frame-shifted spike peptides were significantly increased in vaccinated mice compared to untreated mice or those vaccinated with Oxford AstraZeneca. So I guess I should explain something else here as well. And that is, what is the frame shifting we are talking about? So let's see. Actually, I have an example here. Here is the example. So this is a messenger RNA. Imagine that this is the messenger RNA, which is in the spike protein. The RNA is composed of small coding sequences. So these are nucleotides, these little letters, AUU, GUA, CUU, these are all nucleotides. What researchers have done for the messenger RNA vaccines is that they have modified some of these nucleotides in a way that the immune system cannot recognize them or cannot break them easily or does not react to them because they look like self protein, for example. This allows these messenger RNA vaccines to be able to enter the cell without being destroyed by the immune system while it is injected or very soon after injection. So what happens is, because there are a lot of modified base nucleotides, especially the nucleotide which is the uridine, which stands for U here. The uridine is made into pseudo-methyl-uridine. It's a synthetic uridine. When the ribosome, and what is a ribosome? A ribosome is a tiny machinery in our cells whose job is to read the messenger RNA to make corresponding protein. When the ribosome passes through runs of this UUU, it slips. It stalls and slips. Now, slipping itself is not a huge problem. It slipped. The problem is that when it slipped, it starts making bad proteins. Those bad proteins can now create autoimmune issues. So see here, imagine this is the same messenger RNA. This is a messenger RNA on which, the same one, on which the ribosome was working. It was reading, making proteins. And when it reached here, the ribosome, it slipped by 1. And as soon as it slipped by 1, it started making, it might even stop here and only make this much of a protein, or it might continue to make a bad protein further. Both are bad. The truncated protein is bad because it would fold in weird ways and can cross-react to our body. And the further movement with a bad protein is also bad because that would also create a bad protein that would cause immune issues. So here is an example. Imagine this is messenger RNA. And this messenger RNA is made up of letters, and letters, 3 letters together are called codons. So here is dog and cat saw big hot sun. So I tried to pick up the words that are all 3-letter words. So then imagine that you are reading it and you are ribosome. And as you were reading, because of the letters that were present here, let's say they were so badly, the font was so bad that you slipped on this W and you did not read the W correctly. So what are you now going to read? You are going to read dog and cat. Then you read S and A for saw, but you could not read W because that was just bad font. And so you ended up connecting SA with B. And as you have to read in the sequences of 3, you are ending up with this SABIGHOTSUN and continue. This does not even make sense. This is entirely a different kind of a world now. That is an off-target protein. It is possible that when the frame shifting occurs, then the ribosome accidentally ends up on a newly formed codon, which may be a stop codon, and the ribosome would stop working anymore and end up with a tiny protein which would misfold in a different way. And it's not necessarily that it would make initial part of the spike protein, or it might continue and make an entirely bad protein. So this is a diagram from this study. So they're saying that normally what should happen is that this is a spike that should be formed. But instead, they observed that spike part of the initial part of the spike, and then weird proteins were made. They called them off-target proteins because of ribosomal frame shifting. Bad proteins were formed. This is not some conspiracy theory. And also, this is not something that is causing damage to everyone. However, remember the study that I did a few days ago where you saw that in those people who are vaccinated, their immune system changes to compensate for certain inflammatory states even after 700 days. Remember the T helper cells have to go down. Sorry, the T helper cell was a different study. Remember in this study, their immune system had to calibrate downwards for cardiovascular protection and brain protection and muscle protection, because vaccine had escalated some parts of the immune system. So maybe that escalation occurred because of this. Maybe that escalation occurred because of something else. And here they were showing that only BioNTech or mRNA vaccine produced this issue, and the untreated individuals who did not have the vaccine, or the one with the adenovirus vaccine, they did not have this problem. So I want to look at this. They also gave the sequence. They actually looked at the sequence. And if you see in this sequence, UAG is a stop codon. UAA is a stop codon. So when the ribosome slips and misreads, it can actually stop there, creating a truncated protein. So I want to quickly discuss what else did they find. They found something interesting. They found that in those mice, they did the study then on the mice as well. They found that in those mice where these off-target proteins were made, they found out that the ribosome slips about 10% of the times. That means it makes bad proteins every 10% of the time. And secondly, what they found was that these mice had a greater amount of interferon gamma released in them. Interferon gamma release actually says that the cytotoxic arm of the immune system is active. They did not observe this activation in non-mRNA vaccinated mice. That means, and they also did not see this observation in mRNA vaccinated mice who did not have the off-target proteins. So that told them that these proteins, the bad proteins, are actually causing the innate arm to activate the T helper 1 pathway, which activates the cytotoxic T cells, which are going to go and kill the other cells. Whose cells are they going to kill? Of course, our cells they're going to kill. It's not a bacteria or a virus. So our cells are going to kill. When our cells are going to be broken down, as I said before, they would open up the self as well, which would cause further autoimmunity. So T helper 1 activation and cytotoxic T cell activation was observed because interferon gamma levels were increased in those mice that had off-target proteins. So can they cause tissue damage? Maybe these off-target proteins are just so different that there is no molecular mimicry, meaning they do not mimic any of our tissue surfaces, and we are fine. Our immune system would come, pick them up, destroy them, and done. No issues. Even form memory against them, but still no issues because they're not mimicking any of our tissues. But in those unfortunate individuals where this off-target protein ended up mimicking some part of our tissue surfaces. Then, when the immune system attacks those little off target proteins, the same immune system would then see the same pattern on some of our cells as well and start attacking those cells, creating autoimmunity and then escalation of immunity. That is called cross reactivity. So, can this cause damage? Maybe there is no damage in some. Maybe there is a damage to any tissue. That could be the muscle tissue, that could be heart tissue, vessel tissue, kidney tissue, liver tissue, pancreatic tissue, brain tissue, whatever. It depends what the mimicking did. So this is the discussion. Thank you very much for listening in. The study is interesting. It is disturbing as well. The link is in the description. The second link in the description is for the study. The first link in the description is for a more layman, friendly article. And I want to read this here as well, because last time when I discussed this, I saw so many of my friends, medical friends, jumping the gun and saying, well, that would only be just one part of the spike protein and it cannot do any immune problems. Look at the article from Cambridge University. This is Cambridge University. They are saying, in addition to the target proteins, these slips lead to production of off-target proteins, triggering an unintended immune response. So maybe we need to go to Cambridge University and correct them. Then similarly, if you see here, such design modifications, so they then offered a design modification to fix this problem in the vaccine. Such design modifications can easily be applied to future mRNA vaccines to produce their desired effect while preventing hazardous and unintended immune responses. Unintended immune responses. When the ribosome is confronted with a string of these modified bases called N1-methyl pseudouridine in the mRNA, it slips around 10% of the time, causing the mRNA to be misread and unintended proteins to be produced, enough to trigger an immune response. Removing these runs of N1-methyl pseudouridine from the mRNA prevents off-target protein production. So this is the discussion. Thank you very much for joining in. Please like, subscribe, and share. There is a set of links in the description where if you wanted to support this work, you can buy me a coffee, or there's Patreon. Become a member of Patreon. I would love if you become a member of Drbeen, and we study medicine over there. So thank you very much. Have a great weekend, and I would see you again.